Hello again, I'm Liz McDaniel, and this presentation is all about how the kidneys work to filter blood. Specifically, it's about a type of filtration called glomerular filtration and the forces which play a key role in this specific process. This is for Biology 520, Human Physiology 2 with Dr. Waller at Grand Canyon University. Okie dokie, right here above me is an image of a kidney. Remember that the kidneys maintain homeostasis by regulating electrolytes and water. They eliminate metabolic wastes, and they regulate blood volume and solute concentration, which is also called osmolarity. Kidneys also regulate pH in the blood by controlling acid-base elimination in the urine, and they produce renin, which is a hormone that triggers a chain reaction within the body necessary for salt conservation, and they also convert vitamin D into its active form. Before I talk about glomerular filtration, I'm going to review the kidney and nephron anatomy, and I'll try to highlight as I go. So each kidney is supplied by a renal artery and also has an afferent renal vein. Here is the renal cortex, which is this outer area all around. And the renal medulla is this middle region here, within medulla meaning middle. And the renal medulla consists of these renal pyramids. They look like little triangles. These areas are the sites of urine formation, which then, of course, drains into the renal pelvis, which is this area right here, where it will be whisked off to the ureter, into the urinary bladder, and excreted through the urethra. Over here is a zoomed in nephron. This is the functional unit of the kidney. Remember, each renal artery branches off into teeny tiny renal arterioles, which supply a single nephron. And there are over 1 million nephrons in each kidney. And then these arterioles culminate into this ball-like tuft of capillaries called the glomerulus. Here is where water and solutes are filtered and fluid is collected into a cup-looking structure surrounding it called a Bowman's capsule. This fluid then enters the tubular system as opposed to the capillary system of the nephron, which consists of a proximal tubule up here, the loop of Henle, which descends down into the medulla, and then back through the distal tubule, where it will be ultimately eliminated as urine. And now that we have the anatomy out of the way, let's talk about the renal processes. I have highlighted in yellow here the 3 filtration processes, which are glomerular filtration, which is the first step in urine formation, in which blood flows through the glomerulus and plasma filters from the glomerular capillaries into the Bowman's capsule. The second is tubular reabsorption, in which filtrate flows through tubules. The necessary ions are reabsorbed back into the blood through the membrane into nearby peritubular capillaries. And finally, tubular secretion, which is the same mechanism as reabsorption, but in the opposite direction. This is the selective transfer of substances from the peritubular capillaries into the tubular lumen. And last but not least, of course, is the excretion process, in which the filtered fluid is no longer tampered with or changed by diffusion or reuptake of any additional solutes. It is carried through the ureter into the urinary bladder, where it would be voided through the urethra. The specific process I'm going to talk about in this presentation is that first filtration process called glomerular filtration. And here is a zoomed in image of a single glomerulus. Remember that the renal artery branches out into many tiny arterioles, which each supply an individual nephron. So here you can see this afferent arteriole flowing into the glomerulus. And the structure is unique because its membrane acts as a kind of sieve that retains blood cells and plasma proteins, but allows water and smaller solutes to filter through. This is because that glomerular membrane is much more porous than other arterial wall structures in the body. It's composed of 3 layers. The first is the glomerular capillary wall, which is a single layer of flattened endothelial cells. These are perforated by many large pores, making it 100 times more permeable than other capillaries in the body. And then next we have this basement membrane right here, which is not cellular. It's actually kind of a gelatinous layer made up of collagen and glycoproteins that fill in the space between the glomerulus and the Bowman's capsule. The collagen provides strength, and the glycoproteins prevent plasma proteins from accidentally filtering through. The only plasma protein that can fit through this layer is albumin, but because of the way albumin is charged, less than 1% of albumin proteins are able to enter into this space. And finally, we have the inner layer of the Bowman's capsule, where the solutes will enter the tubular system of the nephron. This layer consists of special cells called podocytes, which have primary and secondary foot processes. You can see these here. Remember the root words pod, meaning foot, and site refers to cell. These foot-like extensions encircle the glomerular capillaries, and they leave small spaces between them called filtration slits. This is a site where excess ions and also waste products from cellular metabolism are able to exit the capillaries, move through that acellular membrane, and enter into the Bowman's capsule, where they will be taken up by the nephron tubular system. Keep in mind also that only about 20% of blood that flows into the glomerular afferent arteriole is filtered in the glomerulus, and the remaining 80% flows through the efferent arteriole into the peritubular capillaries, where it can be picked over for remaining necessary ions as needed to maintain proper salt and ion balance. And that occurs due to those other two filtration processes I mentioned in the previous slide. Just like in other artery and capillary systems in the body, pressure is the primary driver of filtration. Of course, the same principles of fluid dynamics apply here as they do in other venous and arterial structures in the body, but there are two differences in the kidney's nephron systems. First, glomerular capillaries are much more permeable than other capillaries, allowing for more fluid to be altered in the diffusion and filtration processes. And second, the balance of forces across the membrane allows filtration to occur along the entire length of the capillaries rather than just at the or mostly at the beginning. In systemic tissues, gas and ion exchange relies heavily on concentration gradients as well, and that plays a role here. But in the kidneys, there are several forces involved also. So the first is called glomerular capillary blood pressure. This is the first and strongest force involved and is most similar to the way blood pressure changes fluid composition in systemic tissues. Notice here how the afferent arteriole is much larger in diameter than the efferent arteriole, which is much smaller. This pressure exerts a force of about 55 millimeters of mercury and favors filtration. The two other forces actually discourage filtration, but they're so small that the net filtration pressure remains positive, so that filtration still occurs. The second force is called plasma colloid osmotic pressure, and this is caused by an unequal distribution of plasma proteins across the membrane. The partial pressure of water is higher on the inside of the Bowman's capsule, right here, than it is in the glomerular capillaries, meaning that the water in these two structures favors diffusion across the membrane into the glomerular capillaries. This is a smaller force and exerts a pressure of about 30 millimeters of mercury. And the third force involved is the hydrostatic pressure of the Bowman's capsule, which is exerted only by the fluid in the initial part of the tubule right here. This pressure also discourages filtration, but is at an even smaller pressure, exerting only about 15 millimeters of mercury. So even though there are forces discouraging filtration within the nephron, the overall net filtration pressure remains positive, because if we subtract the plasma colloid osmotic pressure and the Bowman's capsule hydrostatic pressure from the glomerular capillary blood pressure, we get a net difference of 10 millimeters of mercury. When we take this net filtration pressure and factor in the effects of glomerular surface area and permeability collectively called the filtration coefficient, we can derive the glomerular filtration rate, which is the filtration coefficient times the net filtration pressure. This overall GFR can change slightly, primarily due to changes in glomerular capillary blood pressure, but other factors such as a decrease in plasma protein concentration or dehydration due to illness can also change glomerular filtration rate. Also, physical blockages in the urinary tract can also negatively impact filtration rate, as can other blockage issues such as a kidney stone or an enlarged prostate. What is really neat about the nephron afferent capillary, though, is that it will adjust to suit the body's needs. So if glomerular pressure rises, these arterioles will decrease in caliber to slow the flow into the nephron. And if systemic blood pressure is too low, the afferent arteriole will dilate to allow a greater volume of blood to flow into the nephron. And that's all I have about glomerular filtration today. I hope you enjoyed learning more about the kidneys as much as I did.